What time is it? It's science and pajamas time. Everyone's favorite time because you get to see everyone's favorite bio teacher. Yes, I know. I gotta say things to make myself feel better. So, last time we had talked about meiosis and how crossing over occurs, but I did just want to kind of go through and make sure that we understand what crossing over is. So, here is one homologous pair of chromosomes. If you notice, they have all the same letters on them, don't they? A through G. But we see capital letters on this one and lowercase letters on this one. They're the same letters, just different variations of the same. And that's what we mean when we talk about alleles. They're just variations of the same gene. There's variations of the eye color gene, which is why we have different eye colors. There's variations of hair color genes, which is why we have different hair colors, and so on, so on. So what happens is that first, if you remember, before we can even go through the steps of meiosis, we have interphase 1, which is when we take our chromosomes our DNA, and we duplicate them. Stupid glares. All right. So now I took my homologous pair and I duplicated them. Once I do that, then in prophase 1, they get super duper close that they form a tetrad. The tetrad is just when they get so close that it allows for something called synapsis to occur. And what that is they get so close, they actually fuse together. And when they separate, they're going to exchange some information. So we end up with I got lots of little pieces down here. So let's say we end up with, so I go bye-bye, and we have that, and then we have that. Now again, we have the same gene still. We just have different combinations of the alleles. Here we have all the lowercase letters. Here we have all the uppercase. Here the first 3 are lowercase, but the bottom 4 are there we go. Are uppercase. Here the top 3 are uppercase, and the bottom 4 are lowercase to allow again for variation. Trying to figure out the best way to move my little thingy because of the sunlight coming from the top window. Let's see. Well, since we're not working in marker, actually it doesn't affect us that much. Oh, nope, never mind. This eye is going to start affecting us. No! Alright, so, there we go. And just get like a box or something to put behind here. Alright, so there's that. That means now, wait a minute. Idea. Won't work for all the times I have to write on this, but it might work for this one. There we go. So it means that once we go through and we do meiosis 1, we're going to separate the 2 homologous pairs. So now these are in different cells. And then after we're done with meiosis 2, we then separate the sister chromatids. Yeah. Hopefully at some point before the quarantine ends, I will get a way to get this taken care of just right. So now we end up with these 4 individual chromatids, and they are each in a different daughter cell, a different gamete at the end of meiosis. Now that means that there are 4 different genetically unique possibilities of this chromosome that can get passed on to the offspring. So from crossing over of one pair, we end up with 4 possibilities. And not just that, doesn't mean that they're always going to fuse and change between the C and D. Maybe just the F and G get changed, or the A and B, or A, B, C, D, and E, F, G travel together. There's different points where they can fuse. So that also brings in even more variation. But we don't have just one of these chromosomes. We got 23. And oh yes, I actually do have enough to show that. So, I don't think I have enough magnets, so I'm going to push this back a little bit. There we go. Alright. So, that means that each one of these is in their own gamete. Now let's add another one. Another example. Let's see, where's my next one? So I really do have a whole bunch of these. I got bags of these things. So I just gotta find the right ones. Okay. So let's say we also have J through O. That's another chromosome. And again, there's different places where crossing over could occur. J, there's J. JKL. 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 There you go. There's those. And here we go. So let's say we also have I'm going to move these to kind of the 4 corners. So we can talk about them a little bit easier. Different possibilities. So this represents the different possible gametes. We have 2 down there, and we have 2 up here. Those are the different possible gametes. The middle is just where I'm storing things. So as we saw before, we go through, we duplicate, we have crossing over going on, and we can end up with maybe something like that, and there we go. It's like a Where's Waldo of chromosome pieces down here. So now we have those 4. Again, did it have to be those 4? No. Maybe only the J swapped during crossing over. Maybe it was the JK. Maybe it was just the O or O and M. There's different ways it can fuse and allow for different variations. Now the next question is, where are they going to go? Well, that's something called random assortment, or independent assortment, I should say. There's 2 laws that Gregor Mendel, which we'll get to in another video, came up with. The first is the law of segregation, which says that the homologous pairs are separated from each other. That we separate all the homologous pairs. And that's what we did up here. So that demonstrates the law of segregation. Then the law of independent assortment says that the different homologous pairs have no effect on how the others separate. So when these guys are going to separate to the different gametes, there's different ways that could happen. So we could maybe have that, and this, and that, and this. That is one possibility. Or we can have that as a 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 possibility. Or that. Or that. That is a lot of possibilities. And how this larger pair separated and divided among the 4 gametes will not affect how any other pair will separate and divide among the 4 gametes. I didn't keep track of the different numbers of ways we could have separated them, but there's a lot, weren't there? And again, I know I've said this in a previous video, that's just with 2 homologous pairs. Again, you have 23 pairs of chromosomes. So imagine that much variation, but then taking it to the idea of 23 pairs with that. So it leads for a lot of diversity, a lot of genetic variation. And that's what we mean by saying that the gametes are genetically identical. They have a lot in common, but they don't have everything in common. So they are genetically unique. All right, so just wanted to review chromosomal separation with you guys and make sure you're kind of understanding what's going on with crossing over. And I hope this helped. Again, if you have questions, concerns, hit me up in our Google Classroom. But until then, until next time, my munchkins, you stay healthy and stay well. Miss you guys. All right, bye-bye.